Okay, today I'm going to start a new topic. My slide is visible.
Yes, sir.
Yes, sir.
Yes, sir.
So today I'm going to talk about the next topic is important molecular process inside the cell, that is replications. So replications, any idea what is replications? Anybody able to define replications?
Sir,
Sir,
formation of
producing
new
new
one
copies, sir.
Yeah.
from existing one, sir.
Actually, replication, it is an important process inside the cell. The entire double-stranded DNA, or in other words, the entire genomic DNA, it will be copied and produced exactly identical to the second copy of the DNA double helix, the entire DNA double helix. So this particular process is unique features of the DNA. You're not able to see in any other biomolecules, RNA or proteins or fat molecules or carbohydrate. Only you can able to see inside in the DNA only. So now, so this slides depicts about the prokaryotic replications. So you can able to see these are the various steps in the prokaryotic replications. First, the prokaryotic parent cells initiate the replications. You know about cell cycle. There are different phases in the cell cycles. First, so whenever the replications will be start, first it will be start from the particular place in the genome. So that particular place you can call it as origin of replications. Okay, but in that particular place, once the replications is going to start. So it will be initiated. So initiated, then it will be the entire genomes will be copied. So entire genetic identical copies of the genetic materials will be produced. So now the particular cells having the two copies, then what happens? The cell size will be elongated and cross walls will be formed, and finally the cell division should be takes place. So a little bit cell biology. I'm not going too much in detail about the cell biology. So from the single cells, you are getting two daughter cells. Why I'm calling two daughter cells? Any idea? So this is the parent cells, prokaryotic parent cells. From the parent cells, two daughter cells will be coming. So two cells will be coming. One cell will be converted into two cells. The two new cells we can call it as daughter cells. Why I am calling daughter cells? What happened parent cell? Any idea? என்ன தம்பிங்களா ஏதாவது சொல்லுங்க.
The daughter cell has the information from the parent cell, sir.
Okay. So one cells will be converted into two cells, right? So,
Yes, sir.
so first initial cell we can call it as parent cells. Now the parent cells will be converted into two daughter cells. So ஒரு செல்ல இருந்து இன்னொரு செல் வருது. அதாவது one cells will be converted into two cells. So once both the cells we can call it as daughter cells. Why? ஒரு பேரண்ட் ஒரு டாட்டர் அப்படி சொல்றாங்கல்ல, ஏன் ரெண்டு செல்ஸுமே நம்ம டாட்டர் செல்ஸ்னு சொல்றோம்?
அது ரெண்டுமே திருப்பியும் ரீப்ரொடியூஸ் பண்ண முடியும், ரெப்ளிகேட் ஆக முடியும்.
That will be self-replicated. It will be replicated. What is my question is, from the single parent cells, two cells will be coming. One cells will be converted into two cells. And the ரெண்டு செல்ஸுமே நம்ம we are called as daughter cells. நான் சொல்றேன், this is parent cell, this is one new daughter cells அப்படின்னு சொல்லாம, why it is called as both the cells is daughter cells? Again, this daughter cells again it will be replicated. Again from the daughter cell, two more daughter cells will be coming. Here also a replication, then two more daughter cells will be coming. Right. So what my question is, what happened the parent cell? Why you are not called as parent cells and daughter cells? Why that both the cells is called as daughter cells?
Replication ஆகுதுனால, sir.
Replication ஆகுது because You can able to see
The genetic information in a bacterial cell is stored in the form of a double-stranded, covalently closed circle of DNA. Replication begins at a specific site called the origin. The origin replicates, and then DNA replication proceeds in two directions. The two original strands, shown as solid lines, serve as the templates for synthesis of new strands, shown as dotted lines. This is referred to as semi-conservative replication.
So, what do you understand from this animation? Hello. யாருமே இல்லையா?
சார் இருக்கும் சார்.
சொல்லுங்க தம்பி. See, initially it started from single cells, right? Two double. This is animations is explaining about the bacterial replications. You know very well the bacterial DNA is circular, so you can able to see the two strand. One is represented in green, blue color. The another strand will be complementary strand will be denoted by this identified. You can able to see in red color. Now during the replications, the both the strand will be act as a parent strand, template strand. Then the complementary strand will be newly synthesized, right? So over daughter cell one strand will be received from the parent cell. The another strand will be synthesized newly. That's why 50% only it will be present from parent cells in both the daughter cells. 50%, that is complementary strand, will be synthesized firstly. That's why we are calling as both our daughter cells, because the complementary strand entirely will be synthesized with the help of the parent template strand. Okay, so if you look at this, so what are the prerequisites for the DNA replications? So DNA replication nadakanuna adukku vandu prerequisites, what are the things means? So must occur accurately, isn't it? Because the DNA will be transferred from one generation to another generation. So without any mutations, without any error, the DNA should be copied perfectly. Because if any error will be there during the replications, it will be affecting the subsequent generations. So that accuracy is very, very important. Then all parts of the chromosome must be duplicated only once. So once the replications is started, so each cycle only one time the replication started. So duplicated only once in one cell cycles to avoid any risk effects, that is harmful effect of gene amplification. Only one time the DNA amplifications will be takes place during the one cell cycles. Then during the cell cycle, that is during replications, the chromatin organizations must also be reproduced, because replication, the DNA compact of the DNA structures will be relaxed, because the association between the histones and the DNA molecules get disturbed. That is, what I'm meaning is the quaternary structures is disturbed. Again, the quaternary structure is formed. So these organizations will be maintained in the daughter cells also. So these things is very prerequisite. If everything will be normal and another, it will be 100%. The daughter cells will be do all the functions properly. So for DNA replication, what are the things is essential? You know that is a template DNA. We need double-stranded DNA. That is a genetic material. That is very essential. Then substrate, you know what is DNTP, deoxynucleotide triphosphate. That is, it is composed of DATP, DCTP, DGTP. All those things will be coming. That is simply you can call it as deoxynucleotide triphosphate. Then we need primer. Primer means that is RNA, mostly DNA replication. We can using RNA primer. That is a short nucleotide RNA polynucleotide with free 3'-hydroxylane. Then we need some enzymes. DNA dependent DNA polymerase. There are so much of enzymes will be involved. We are going to discuss one by one. So in this slide, just these things you have to very, very essential. Template DNA is essential, and substrate, primer and enzymes, and some protein factors. There are number of protein molecules will be playing a essential role in various stages of the replications. We are going to discuss all those things. So, what are the features of the DNA replication? Some important things. DNA replications is semi-conservative. It was discovered by Measles and Stogel. Experimentally, these people proved DNA replications by semi-conservative method. There are three hypotheses will be there earlier. Conservative method, dispersive method, semi-conservative method. So these people, Measles and Stahl, 1958, experimentally proved out of three hypotheses, DNA replications by semi-conservative method. We will discuss later all those things. Right? So replications require polymerase enzyme. We can call it as DNA polymerase. For RNA synthesis, RNA polymerase. So DNA polymerase, and already we mentioned template and primer and nucleotides. So always the directions of the synthesis of new strand always from 5' to 3' direction. You know why it is called as 5', when it is called as 3'. The end of the nucleotide 5' the fifth carbon level, the sugar level, the phosphate group will be 3. That is 5' end. Third group level, the hydroxyl group will be free. That is 3' end. So this information is given by the Korenberg in 1957, right? So replications in E. coli genome, there is, you know that each and every cells of E. coli having only one copies of chromosome, single chromosome, that is circular DNA. So it will be stored at the specific site in the genome. It won't start randomly. In order to start the replications, it will be stored at the specific site. That particular site we can call it as ORI, or origin of replications. In E. coli genome, you can able to see only one origin of replications. In our cells, you can able to see number of origin of replications. Okay? E. coli only one origin of replication. In our eukaryotic cells having number of origin of ORI will be there. So replications, the movement of the replication, the synthesis of the replication, the synthesis of the complementary strand is bidirectional. It will be move in both the directions from the origin of replication. Suppose origin of replications in the center, either side the replications will be takes place, both right hand side and left hand side. That's why it is called as bidirectional replications. So these things will be given by the Keynes 1963. The replication is semi-discontinuous. It means that you know very well there are two double helical structures, two strands will be there. One strand will be going 5' to 3' direction. The complementary strand will be going 3' to 5' directions. The new strand will be synthesized. That is a already existing parent strand. It will be used as a template strand to synthesize the complementary strand. So one strand will be synthesized continuously without any disturbance. That is, we can call it as leading strand. Continuous strand is otherwise called as leading strand. The another strand will be discontinuously takes place. That is, we can call it as lagging strand. இது ஏற்கனவே படிச்சிருக்கீங்களா எங்கயாவது? Students, already studied about leading strand, lagging strand, about the replications anywhere?
No, sir.
This is very new to you, new information.
Yes, sir.
Asking everyone, anybody studied?
Sir, 5' to 3' leading strand, 3' to 5' lagging strand. படிச்ச மாதிரி ஞாபகம் சார்.
எங்க படிச்சிருக்கீங்க?
12th சார்.
12th. ஓகே ரைட். இருந்தாலும் கொஞ்சம் படிச்ச மாதிரி ஞாபகம் இருக்கு. இப்போ நான் வந்து உங்களுக்கு டெக்ஸ்ட் வச்சு பேசிட்டு இருக்கேன். Next I will going to show the pictures. Okay, this is the important features in the DNA replications. We are going to highlight the features in the DNA replication, then we'll go one by one, right? So don't worry about that. என்னடா ஒரு டெக்ஸ்ட் வச்சு பேசிட்டு இருக்காரு, தூக்கம் வர்ற மாதிரி நினைக்காதீங்க. Just I'm going to show the pictures soon. The important features in the DNA replication I try to highlight it here. So another strand will be synthesized semi-discontinuous. It will be because it will be synthesized in the form of fragments. That's why it is called as lagging strand. So both synthesis of semi-continuous and discontinuous requires RNA primers. Okay, this information is given by the Okazaki in 1968. Okay, so because that is the fragments, it will be synthesized in the form of fragments. That fragments we can call it as Okazaki fragments. And the scientist பேர்லே வந்து அந்த fragments சொல்றோம். Okazaki fragments. So there are 3 polymerase will be there in the E. coli. Out of 3 polymerase, DNA polymerase 3 is the replicative enzyme for E. coli. The main enzyme. E. coli DNA polymerase 3 is the main enzyme for the replications. This is given by the Keynes and the Lucia in 1969. So these are some of the important features in DNA replications. And replicons means any segment of the DNA, any piece of DNA which replicates as a single unit, we can call it as replicon. For E. coli, for example, like E. coli, only one origin of replications will be there. So yeah, all the at a time, the entire genetic materials will be replicated. So you can call it as one replicon. That is, the entire things we can call it as replicon. So in E. coli, its origins will be there where the replication is start. We can call it as origin, and terminus where the replication end. That we can call it as terminus. Okay. So in eukaryotes, I already informed that the number of origin of replications will be there. E. coli, one origin of replication, one termination site. In eukaryotes, there are number of origin of replications will be there, but each and every origin is not end up with the termination site. Okay. There are multiple origin of replications will be there. That's why it is called multiple replicons in eukaryotes genome. Bidirection I already informed. Okay. All prokaryotes, bacteriophage, and viral DNA molecules are circular and having only one replicon. So exactly opposite 180-degree angle, you can able to see this of the termination site. That is a ter site. Termination sites will be present in the bacterial systems. So eukaryotes multiple replicons. That is because the multiple origin of replications will be there. You can able to see that 50,000 to 1 lakh replicons will be there in the mammalian cells. Each replicon is spaced in between gap, that is a 550 to 300 kb apart. So one replicon is length on the 50 to 300 kb. Next replicon after the 300 kb, next replicon should be there. So you can able to see number of replicons. First time yeast that is a lower eukaryotic cells. First time they are identified the eukaryotic systems having multiple replicons. So each and every replicons will be expression patterns will be differs from the same cell, same stage, other various stages and various tissues. The expression patterns of the replicons will be differs, but the replication speed will be same. So I already informed that there is no separate terminal site for the each and every replicon in eukaryotes. Okay. So look at this. This slide is explaining about the different steps. First, it will be recognizing. The origin of replications will be recognized by the initiator. Okay. Once it will be recognized, the origin of replications, then it will be melted. It means that in that particular regions, the double helical structures will be disturbed. That is unwinding. Melting means the hydrogen bond will be broken down. That's why it is called as melting. Okay, the hydrogen bond will be broken down. Then helicase enzymes will become and unwind. For zip open mandra mari, it will be opening. Then the DNA polymerase, all the protein factors will be bind, and you can able to see small beads here. You can able to see here small beads. That is called as SSB. That is single stranded DNA binding protein. So it will be bind only with single stranded DNA. It will be prevent the renaturations of this complementary strand. So what happens? First thing is I already discussed the multiple linear origin of replication first identified in budding yeast, low eukaryotic organisms. Okay, so you know very well which phase of the cell cycle the replication starts. Any idea? I already studied about the cell cycle in cell biology.
S phase.
S phase. When it is initiated, it means that S phase only the DNA start replicating, right? That is synthesis phase. But actually the process will be started little bit earlier.
G.
Huh? Actually little bit earlier.
G
But
phase, sir.
G1 phase.
Yes, sir. G phase, sir.
Yeah, G phase, G1 phase. That is a late M phase and G1 phase. That is a transient phase. Actually, adukana steps ஆரம்பிச்சிரும். But exactly the DNA duplication should be takes place only in the S phase. That's why it's called a synthesis phase. But the process for the replication start little bit early. Okay. So we'll see. You can able to see here. This is this pictures is giving some explaining about the cell cycle and what are the regulatory molecules will be involved. So with help of the growth factors, you know that this is G0 phase. When the cell cycle will be started to G0 phase, this is a resting phase. So resting phase will be kind of entered into the G1 phase. It will require some cyclin D. Okay. This cyclin D will be synthesized by the growth factors will be triggered, and these are all the pathways. I am not going into detail about this pathway because it's entirely cell biology. Just I'm going to highlight. So these cyclins will be synthesized, then it will be activating the CDK, right? CDK4 and 6. So it will be used to cross the restriction point of the G1. Okay, these things is very essential. Then if you want to cross from G1 to S, if you want to enter into the S phase, then CDK2, this cyclin E and CDK2 will be activation. With help of the cyclin E, the CDK2 is activated. Then it can able to cross this checkpoint. So each and every checkpoint, some checking will be takes place. Each stage of the cell cycle, everything will be going in proper way or not. Then if it is that, then it will be entered into the synthesis phase. Here exactly this synthesis of the that is DNA will be duplications will be takes place. In previous stage, the helicase everything will be attached in the origin of replication, but it will be inactive form. Inactive form. So any things will be cross this restriction point. Either cross IT, cross IT S குள்ள போயிடுச்சுன்னா then அதுல எந்த ப்ராப்ளம் இருந்தாலும் வந்து synthesis phase halt ஆகாது, synthesis phase நடக்கும். இங்க தான் செக்கிங் நடக்கும். இங்க வந்து இந்த செக் போஸ்ட்ல இருந்து எஸ்கேப் ஆகி இங்க வந்துச்சா திருப்பி இது வந்து இம்ப்ராப்பரா இருக்கு, இங்க நியூக்ளியோட்டிக் சீக்வென்ஸ்ல ஏதோ ப்ராப்ளம் இருக்கு, அகைன் பின்னாடி போகாது, not able to go reverse way. Then it will be leads to cancer. This is the way the cancer should be developed, because the problem in the checkpoint proteins, restriction point regulator, all those things. So in S phase, then it will be entered into the G2, again the cyclin A and CDK1, then it will be able to cross this M checkpoint, that is a G2 M checkpoint. Again, this is CDK1 cyclin A, it will be completely, it will be, mitosis will be end, and again, next cell cycles will be start. This is going on. Okay. So I already mentioned the initiations of the replications divided into two steps. So actually it will be started late M and early G1. Okay. It started very early, but activations of the replications will be takes place in the S phase. It will be an inactive form. The pair of replicative helicases MCM, it will be all that is helicase, the enzyme will be used to unwind the DNA. Everything will be identify the origin of replications, will go and bind in this regions with help of the pre-replicative complex. It already go and position properly. So இதெல்லாம் கரெக்ட்டா வந்து உட்கார்ந்தாதான் then the checkpoints இருக்குல, அந்த checkpoints வந்து it will giving license for the replications. Okay, you can proceed for the replication. You can able to enter into the S phase. I already informed there are number of origin of replications will be there in the eukaryotic organisms, but the expressions of the origin of replications, it will be differed from tissue to tissue and different stage of the metabolisms. So in the late early G1 phase la vandu endha origin la vandu the helicase enzyme and that is a pre-replicative complex will be placed properly, then it will be screened by this restriction point, then allowed to enter into the S phase. So அந்த origin மட்டும் தான் வந்து ஆக்டிவ் ஆகும். எந்தெந்த ஆரிஜின்ல எல்லாம் வந்து இந்த என்சைம் ரெப்ளிகேட்டிவ் காம்ப்ளெக்ஸ் வந்து பைண்ட் ஆகலையோ அந்த ஆரிஜின்ல எக்ஸ்பிரஸ் ஆகாது. ஓகே, அகைன் தட் அந்த ஆரிஜின் எக்ஸ்பிரஸ் ஆகணும், திருப்பி நெக்ஸ்ட் ஒரு ரவுண்ட் முடிச்சு திருப்பி மைட்டாசிஸ் முடிஞ்சு திருப்பி G1ல வரும்போது தான். ஓகே, இட்ஸ் ஒன்ஸ் started, it's not going back in reverse directions. This is termed licensing of replications. ORIS initiation S phase can occur only at those origin that have a pre-RC. எந்த ஆரிஜின்ல வந்து ப்ரீ ரெப்ளிகேட்டிவ் காம்ப்ளெக்ஸ் வந்து பைண்ட் ஆயிருக்கோ அந்த ஆரிஜின் மட்டும் தான் வந்து எக்ஸ்பிரஸ் ஆகும். So once it we enter into the S phase, previous stage it's already bound in this origin of replication but is in inactive form. Once it we enter into the S phase, DNA helicases are activated. For activation required some protein. We will discuss. So what happens once it will be activated, the helicase will be unwind the DNA. It will melt the hydrogen bond and the double helical structures will be converted into single stranded. That is unwinding, bulging. Okay, initiations of the DNA synthesis, the helicase moves out of the origin. So they start moving the helicase from the origin of replications. Unwind பண்ணிட்டு அப்படியே வந்து it will be going in forward directions. You know that from the origin of replications, there are two helicases will be bind. One is going in a clockwise direction, another one is going in anti-clockwise direction. Bidirectional replications. See, the origin site now lack the bound helicase and cannot reinitiate replications until after it bind again in the next G1. So hence, origin can be activated only once per cell cycle. Right? It will be activated. It will be the pre-replicative complex will be formed in the late M and early G1. So look at this. So this is the origin of replications. Okay, that is ORI. The ORC, this is the origin recognizing complex. It's a multi-subunit protein. It will come and bind. In general, the expressions pattern of the CDC6 okay, will be very, very less. But during the initiations of these replication, the concentrations of the CDC6 will be more. It will go and bind with the ORC. Then again, with the help of the CDC6 and CD1, it will be bringing this helicase enzyme into this origin of replications. So these things will be takes place in the early of S phase. CD6, CDT1 and ORC. These proteins will be brings these MCM helicases. That is helicases. That is a mini chromosome maintenance proteins. It will be helicases activity will go and come and bind in these regions. Okay, now it is called as pre-replicative complex because ORC along with CD1 and CD6 MCM helicase. I already mentioned two helicases. One is going in this direction, another one is going in this direction. So now this origin is licensed. In the origin the replication start ஆகலாம்னு ஒரு லைசென்ஸ் கொடுக்குது அந்த ரெஸ்ட்ரிக்ஷன் பாயிண்ட்ல அந்த ஓகே. ஸோ இந்த ஆரிஜின்ல வந்து ஆக்டிவா இருக்கும். அடுத்தது எஸ் பேஸ்ல வந்து இந்த ஆரிஜின்ல இருந்து வந்து ரெப்ளிகேஷன் ஸ்டார்ட் ஆகும். ஸோ இதே மாதிரி ஐ ஆல்ரெடி மென்ஷன் தெர் ஆர் மல்டிபிள் ஆரிஜின் இன் தி என்டையர் வேர்ல்ட்